Welcome to The Explainer. Today, we're cracking open the immunologist's toolbox. We're going to find out how scientists manage to make the completely invisible world of our immune system, well, visible. We'll explore the incredibly clever tricks they came up with to watch a war happening on a scale that's almost impossible to imagine.
Right, so think about this. Every single second, there's a silent war raging inside your body between your immune cells and trillions of potential invaders. But this whole battle is fought by molecules and cells, things so tiny you can't really see the whole picture. So the big question is, how on earth do you study a fight that you can't even see?
Well, the first brilliant idea was to create a kind of custom-made searchlight that could find just one specific enemy, or as scientists call it, an antigen. So they immunize an animal with that one specific target. The animal's immune system then becomes this tiny, super specialized factory, churning out millions of antibodies. You can think of them like little molecular detectives, each one built to hunt down and grab onto one and only one target.
But of course, there's a catch. Just injecting an antigen usually isn't enough to get a really powerful response. The animal's immune system might see it, sure, but it might not think it's a big deal. So you only get a trickle of those antibody searchlights we need. To really light up our target, we've got to find a way to sound a much, much louder alarm.
And they do this using something called an adjuvant. Honestly, the best way to think of an adjuvant is like a hype man for the immune system. You mix it in with the antigen, and its whole job is to basically shout, "Hey, over here, pay attention to this thing." And that makes the immune system launch a much stronger, more focused response, which means we get exactly what we want, a massive supply of those super specific antibodies for our experiment.
And what's really cool is how these adjuvants work in different ways. Some of them, like the famous Friends adjuvant, create this little oily pocket that acts as a depot, just slowly leaking out the antigen over time. This keeps the immune system interested for an incredibly long time. We're talking up to 544 days in some studies. Others, like parts of the bacteria that causes whooping cough, just directly supercharge the very immune cells, the B cells that make the antibodies.
Okay, so we've used our adjuvants, we've got a ton of custom antibodies, and we've put them in a sample to hunt for their target. So now what? The next big hurdle is, how do we actually see the moment of detection? How do we know when our little searchlight has actually found what it was looking for?
Well, this leads us to a truly foundational discovery called the precipitin reaction. See, when enough of our antibodies, which have two hands, by the way, find and grab on to their target antigens, they don't just stop there. They use their second hand to grab onto other antibody-antigen pairs, linking everything together into this huge, interlocking molecular net. It's really just like weaving a net. A few loose strands of thread are basically invisible, floating in water. But as you start weaving more and more of them together, you eventually get a solid net you can actually see. It's the same idea here. This molecular lattice just keeps growing until it's too big and heavy to stay dissolved, and it literally falls out of the liquid as a visible, cloudy solid. And just like that, the invisible has become visible.
But, and this is a huge but, there's a critical catch. This whole thing completely depends on getting the ratio just right. You absolutely need the perfect balance of antibodies to antigens to build that big, strong interlocking net. If you have too much of one or too much of the other, the whole thing just falls apart.
It's basically a Goldilocks principle. Check out the first bar. You've got way too many antibodies. There just aren't enough antigens to link them all together. Now look at the third bar. You're swamped with antigens. They grab onto every single hand of the antibodies, leaving no free hands to link up. It's only in the middle, in what's called the equivalence zone, that the ratio is just right for building that massive net and getting a big, visible result.
So this precipitation reaction was a total breakthrough, but doing it in a test tube was, well, it was often messy and kind of hard to read. Scientists needed more precision, and their brilliant solution was to move the whole reaction into a brand new arena, a semi-solid agar gel. See, in a gel, you can put your antibodies in one little well and your antigens in another. Then they just slowly spread out or diffuse through the gel towards each other. It's like two crowds walking towards each other across a big field. They're guaranteed to meet somewhere in the middle. And right at that meeting point in the gel, you hit that perfect equivalent zone concentration, and bam, a sharp, clear line of precipitate forms, so much cleaner, so much more elegant.
And this one simple change led to two really key techniques.
The first one, outralani diffusion, is all about identification. You can think of it like an immunological police lineup. It's a detective's tool for comparing different antigen suspects to see if they're the same or related or totally different. And reading the results is like cracking a secret code, but it's beautifully simple. If the precipitation lines form a smooth, perfect arc, that's a pattern of identity. Your antigens are identical twins. If the lines cross right over each other, they're total strangers. And if you get this little spur, it means they're related, like cousins. They share some features, but one of them has an extra unique part. It's just a really elegant way to see molecular relationships. But, just identifying a substance is only half the story. What if you need to know not just if it's there, but exactly how much of it you have? You need to go from a simple qualitative yes or no to a hard quantitative number. And that's where rocket electrophoresis comes in. For this, the antibodies are mixed evenly throughout the entire gel. Then use an electric field to pull the antigen samples through it. As they travel, they react and precipitate out, forming this cool shape that looks just like a rocket. And here's the key insight. The height of that rocket tells you exactly how much antigen you started with. A taller rocket means more antigen. Simple as that. Oh, and it's also about 10 to 20 times more sensitive. So, how does all this elegant science stuff from the lab bench actually translate into tools that affect our lives? Well, let's look at one of the simplest yet most powerful applications of this whole idea, something called agglutination. The concept behind agglutination is just brilliant. Instead of dealing with invisible free-floating antigens, you first stick them onto the surface of something much bigger, like a red blood cell or a tiny little plastic bead. Now what you have is a liquid solution filled with these coated particles, all floating around separately, looking perfectly smooth.
Now for the magic. You add the specific antibody. And just like before, it's two hands grab onto the antigens, but this time, it's grabbing antigens on two different beads, acting like a piece of molecular glue. It pulls those beads together, which then pull other beads together, and within seconds, you get these big, visible clumps. The smooth liquid suddenly looks all grainy. It's a fantastic, simple, yes or no answer. Clumping means you've got a match.
And don't let that simplicity fool you for a second. This technique is ridiculously sensitive. It can detect antibody concentrations as low as 0.001 micrograms per milliliter. I mean, that is an unbelievably tiny amount, like finding one specific needle in a whole barn full of haystacks. It's a total powerhouse tool disguised as a simple clumping reaction. So what this all means is that this beautifully simple principle is the engine behind tons of the rapid diagnostic tests we rely on every single day. The next time you hear about someone getting their blood typed before transfusion, that's an agglutination test, using their own red blood cells as the beads. When doctors need to quickly figure out what specific bacteria is causing an infection, they're often using a test just like this. It's fast, it's sensitive, and it has saved countless lives.
And that's really the journey, right? All the way from a slightly cloudy liquid in a test tube to tools that are absolutely essential to modern medicine. These foundational techniques gave us our very first real window into the immune system's invisible war. It really makes you wonder, what new principles are being discovered right now, and what invisible battles will they let us see next?